Thank you, Doc. I'm feeling pretty inspired this morning from those two previous speakers. So I hope that they've given you a little bit of inspiration. I'll give you a little bit of perspiration. Okay. So I'm going to be talking to you about an introduction to plant pathology and plant diseases. And what I'm wanting to do this morning is to give you a little bit of a background so that the presentations that will come after tea will be much more specific in addressing some of our major plant pathology challenges. So what I'm going to do is give you some background into plant diseases and specifically into the field of plant pathology. So if I can begin by introducing to what is plant pathology itself. So it's a science, and it's the study of the organisms and the environmental conditions that cause plant diseases. I think all of you as budding farmers here, which is our Farming 101 objective, is to introduce you to some of the concepts in farming. I think you will all, in your work, will come into contact with plant diseases in the future. So what are the organisms and the environment that cause these? So we're looking at the pathogenic organisms. What effect does the organism actually have on the plant? So what is that interaction between the plant and the organism? And specifically looking at the environment that's involved in that interaction and how that influences the diseases. And then very, very importantly, of course, is our major objective then is how do we manage those diseases? What can we do? What actions or strategies can we put in place where we can manage those particular plant diseases? Now, if we look at what are the major causes, the pathogenic organisms, so a disease is caused by a specific type of organism, a pathogen. So those pathogens can be fungi or oomycetes, bacteria, viruses, and in addition to that, we can also get mycoplasmas and phytoplasmas. Now, these mycoplasmas, phytoplasmas, these are microscopic organisms that will cause plant diseases. If we look at the plant cell there, I've just got a schematic drawing of a plant cell, you will see the relative sizes of those particular organisms. If that's an entire plant cell, you see we go from the fungi to the bacteria to the viruses, so getting smaller and smaller in the organisms that we're talking about. Very importantly, probably the first thing you will see in dealing with a plant pathogen, you will see the symptoms. So you will see the expression of that disease. The pathogen has caused that disease. The plant now is showing symptoms of the presence of that particular pathogen. So those symptoms can range from the above ground symptoms of your blights, your fruit blemishes and blotches, leaf lesions and symptoms on the leaves, cankers on the stems and the branches, wilting or verwelking, so wilting of your above ground plants. But then it can range right to into the crown area where you can get galls, or you can get your root diseases. And what will happen after tea this morning, my colleagues will then be telling you about some of those important symptoms and plant pathogenic organisms that cause those particular symptoms, either from going from the root systems to the above ground parts. We will be telling you more about those. Now, very importantly, when we talk about plant pathogens and plant diseases, is the concept of the disease triangle. And what we mean by the disease triangle, that is, in order for any disease to happen, or for an epidemic to actually occur, what we need three essential factors. We need a susceptible host, we need an aggressive or virulent pathogen, and we need an environment in which that host and that pathogen will interact and cause the disease. If any of these factors are missing or not appropriate, the disease will not happen. And that for us is very, very useful to know, is that if we can make, say, an environment not conducive to a disease, we can stop or limit a particular disease. And going in connection with limiting that disease, how are we going to manage a particular disease? A quotation that I really, really enjoy, because I'm really involved in disease management, is the concept of what gets measured gets managed. And that's a phrase that has been attributed to Peter Drucker, who was a management consultant. Okay, so again, what gets measured gets managed. And for us as plant pathologists, then we ask ourselves, what can we measure in order to manage plant diseases? And that brings us to the very important science of plant disease epidemiology. When we're talking about epidemiology, very similar to the epidemiology of our human diseases or our human pathogens, the concepts are very similar. But we now, of course, are talking about specifically epidemiology of plant diseases. Now, what the definition of epidemiology is: the study of the development and spread of disease in populations, as well as all the factors that influence the study and development of that particular disease. Okay, probably the most famous plant epidemiologist, it was Dr. J.E. van der Planck, who is a South African. He was a potato breeder and passionately involved in the science of potato diseases. How do they spread? How do they develop? And particularly, of course, how can we manage those particular potato diseases? He came up with two phrases. He said that you can classify plant pathogens into either monocyclic pathogens or polycyclic pathogens. And what I will do then is tell you a little bit about that. If we look at a pathogen and how it will cause a disease, invariably that disease process occurs in a cyclical manner. So within any one season of a crop, you will get a seasonal cycle of that pathogen within that season. So you will start off, there will be inoculum of your pathogen. The inoculum will infect your host. It will then grow in that host or proliferate or colonize the host. Once that colonization has happened, then it will start to produce inoculum of itself. It will sporulate. Okay, so now in a monocyclic pathogen then, what will happen? That inoculum will be released and it will be then spread into the environment, and then probably the crop will be harvested, and then that inoculum that is produced will then be available for the next season. In polycyclic pathogens, on the other hand, what will happen that is within seasons, there can be many, many generations of that pathogen. So the pathogen, the inoculum, the initial inoculum will be there. It will infect the host. It will then colonize the host. It will start to sporulate. But then within that season, that inoculum, that spores that are produced, will go and infect more of the plant. Okay, of that same host, and you can get many, many cycles, and that is then how a polycyclic pathogen works. So that within seasons, there's many generations of that particular disease. Okay, now if we look at polycyclic pathogens, these are your pathogens that really have the explosive diseases. So those are your pathogens in which, as I said, there's many cycles of the pathogen in one season. Now the number of cycles that will occur in that season will very much depend on the interaction between the host, the pathogen, and the environment. Okay, so we're back to our disease triangle again, so that we know that if a particular pathogen, if the conditions are ideal, you're going to get many, many cycles happening of that pathogen. And for us in the deciduous fruit, two of the types of pathogens that are classical polycyclic pathogens are your powdery mildews and also apple scab, are classical polycyclic pathogens. Both of them can have many, many generations of the pathogen in their planting season. Now, if we look at the epidemiology of your polycyclic pathogen, what we see, if we measure the amount of disease, so the amount of disease over time, we then get to draw what is called a disease progress curve. So your disease progress curve, how does that disease develop over time? And the type of disease progress curve you get with a typical polycyclic pathogen is what we call a sigmoidal disease progress curve. In a sigmoidal disease progress curve, what we see initially, we see a very slow development of the disease. And that is because that there is very little initial inoculum there. Okay, so it's a slow initially. Then all of a sudden, when there is a build-up, now I've told you that there's many generations of your pathogen in that season, a build-up of that inoculum, lots of nice susceptible host material there, you get the exponential or logarithmic phase of that pathogen. Okay, that's when it becomes explosive. And then with time, of course, when there gets less and less of the of your susceptible host material, you get then again a plateau region, a slowing down of your disease progress curve. Now, why I give you this, so what I've shown you there then is the sigmoidal disease progress curve. Now, we can use that information then to the very clever people or the mathematicians then can, from that disease progress curve, they can draw then what we call is as an exponential equation. The exponential equation that describes that sigmoidal disease progress curve that we've drawn there is xt, so the disease at a time t is equal to xi, which is your initial inoculum, how much initial inoculum are there, and then R and T. R is the rate at which that disease will progress, and the time that is allowed for that disease to progress. And what you see is the little symbol E, that is the exponential constant. So it's a constant that shows you that that disease is explosive, it's exponential in growth. Okay, so this is a very important concept for us as plant pathologists, because what it means is that we know in order to manage a certain disease, if we can then limit the amount of initial inoculum xi, if we can slow down the rate of the disease progress, or if we can shorten the time of that epidemic, we can then manage that particular disease. And that is why epidemiology is such an important science in disease management, because it enables us to understand why we do what we do in terms of disease management. With that then, that brings us back to the disease triangle. So if we know, if we've got a polycyclic disease, that we need to reduce the initial inoculum, okay, xi, we need to slow down the organism, we need to slow down the development of that disease, okay, or we need to reduce the time. What are our management strategies that we can actually put into place? So we can look at, if we, in terms of that disease triangle, then we can look at the host itself. Okay. The first thing that we can do, of course, in terms of the host, is we can select resistant cultivars, or we can encourage or promote the breeding of resistant cultivars. Because as soon as we actually bring in resistance into our host, either we will not get disease at all, or we will get a very much slower progress of that disease in that crop, okay, by breeding for resistance. In terms of the pathogen itself, if we want to exclude that initial inoculum XI, so a method of excluding initial inoculum, of course, would be your quarantine systems. And that's why for us in South Africa, it's really, really important to have strong quarantine regulations for bringing material into South Africa, because that will stop the import of pathogenic organisms, or it will manage the import of those pathogenic organisms to see, are they putting our industries at risk? Okay. If there is already inoculum there, it's present already, how do we reduce that initial inoculum? So by reducing our initial inoculum, we can have strategies like sanitation, where we will go in, we can remove infected material. We can go in, we can collect leaves that are infected. We can grind them up. We can remove that inoculum from the system. Or if it's a soil-borne disease, is it appropriate to use something like a soil fumigation or soil solarization to remove that inoculum or reduce that inoculum from the soil? And then, of course, very importantly, in terms of our reducing that inoculum there, is our application of appropriate chemicals. So what chemicals can we use in order to limit the amount of initial inoculum that's there? Okay. And then, of course, in terms of the environment itself, there are strategies that we can put in place that directly influence the environment in which our host and pathogen can interact. For example, if we plant our veggies and we plant them too densely, what happens is that we provide the ideal environment in which those pathogens that will occur on the vegetables, and say on the little seedlings that are growing high density, to spread from plant to plant. Because we've increased the humidity in that area, and also by having very dense plantings, if we're putting on a chemical, are we putting it on properly? Can we get a good enough cover? So by improving our plant densities, we can then also reduce those conducive environments. In terms of things like you're managing your trees, very importantly will be your concepts of pruning. So opening up the canopies, allowing the air movement through the canopy, allowing sunlight to get into your canopies, but then you're reducing the humidity within those that environment. By reducing the humidity, it's less conducive for many plant diseases. And I think what I've got to emphasize here is the beautiful link between your pathogen and the appropriate environment. Many pathogens will only be able to infect or to sporulate under very specific environmental conditions. By us knowing and understanding that relationship between the pathogen and the environment, we can then actually manage that pathogen much more effectively. An example would be then in understanding our environment and a particular pathogen, we can then predict whether a disease is going to occur or not. And by being able to predict for something, we can then put early management strategies in place. My last slide that I have then is a summation slide of polycyclic pathogens and how we can bring our management strategies together to manage a certain disease. What I'm showing you here, the curve A would be your sigmoidal disease progress curve of a polycyclic pathogen with no management strategies that have been implemented. If we implement a management strategy that removes the initial inoculum, so we're removing that source of the pathogen, we're lowering that source of the pathogen, what we do is we shift that curve to the right. You see, we've delayed the start of the epidemic. by simply removing the initial inoculum. Okay, so we've shifted the graph to the right to curve B. If we implement a strategy that slows down the rate of development of a particular disease, say we put a fungicide on, or we bring in a resistant cultivar. So what we're doing, we're slowing down the development of that disease within that particular population. What we do, we flatten the curve. So that would be the disease progress curve C. We're slowing it down, we're flattening that curve. What we want to do, ideally, we want to do both of those. We want to combine B and C. So if we can remove inoculum, remove your mummies on your trees if you've got a problem with monilinear luxa, that is an important source of inoculum. If we can really, really do a diligent sanitation program, removing that inoculum, if we can put appropriate fungicides on at the right time, we can then end up with curve D. So curve D then shows you what we call is an integrated system, where we've now used two strategies in order to manage that disease. Okay, and that's ideally what we're looking for. And that for us as plant pathologists that are involved in disease management, we want to be able to integrate as many management strategies as possible. So be that cultivation, biological, host resistance, chemicals. We want to use as many tools as possible, bring them together to manage a specific disease. And so we want to manipulate that pathogen population on that host so that we don't have economic losses because of that particular pathogen. And then, of course, ideally what we would like to also, of course, is reduce our dependence on chemicals. Very important for our strategies that we're putting in place. And I see my time's up, and that's my story. Thank you.